Hello, my name is Hashim Ash, and I'm going to be talking to you today about disordered development of the eyes in oculocutaneous albinism. The learning outcomes for today. After completing this lesson, students should be able to do the following: understand normal development of the eyes, understand the regulatory, genetic, and signaling pathways in normal and disordered development, as well as understand disordered development of the eyes in oculocutaneous albinism. Definitions: retinal pigment epithelium, RP, layer of cells located between the neural retina and the choroid of the eye. Melanin is pigmentation derived from the molecule tyrosine in a reaction catalyzed by the enzyme tyrosinase. Melanocytes are melanin producing cells found in the epidermis, but the iris as well. Foveal hypoplasia is a lack of a foveal pit. A foveal pit is a depression in the fovea, commonly seen in people with albinism. Now for normal development of the eye. The neural tube begins to form around 22 days of gestation in humans. Optic grooves form in this period and become the optic vesicles. These vesicles have contact with the surface ectoderm, which induces the formation of the lens plateau to a variety of genetic and signaling pathways we'll talk about later on. The proximal portion of the vesicle forms the optic stalk, while the distal portion is invaginated to form the optic cup, which then forms the retina, iris, and ciliary body. And a short video.
The first step in visual development is creating the various structures of the eye which make seeing possible. Eye development follows a common pattern in all vertebrate animals, including fish, birds, and humans. As we will see in this fish embryo, the eyes are made early in the developing vertebrate. Back when the body of the embryo is just a long tube of cells, the brain begins to form at one end of the tube. Soon afterward, two smaller tubes grow outward from the developing brain. These tubes, called the optic vesicles, begin to make the basic structures of the eye. We can watch the optic vesicles grow in a developing chicken embryo. Where the vesicles come into contact with the outside wall of the embryo, a layer of cells will fold, eventually becoming the neural retina. At the same time, the lens of the eye begins to take shape from a thickening of the outer layer.
Okay. Now to talk about a few structures in the eye, we'll start with the cornea. The cornea is made up of epithelium from the surface ectoderm. By 8 weeks of development, the cornea has five distinct layers. Corneal stroma, which is the thickest part of the cornea, and the endothelium are both derived from neural crest cells. Mature transparent cornea is a result of mesenchymal stromal keratocytes. Now for the iris and ciliary body, both are a product of anterior rim of the optic cup. The muscles of the iris are derived from neuroectoderm. Neural crest cells produce stroma of iris. Concentration of melanocytes determine color. It is these melanocytes which produce the melanin. Now for the embryologic origin of other major eye structures, the eyelids are derived from the surface ectoderm. Corneal epithelium is derived from surface ectoderm. Corneal, stromal, and endothelium are derived from neural crest. Sclera is derived from the neural crest. Iris epithelium is derived from neural ectoderm. The iris stroma is derived from mesoderm and neural crest. The lens is derived from the surface ectoderm. The optic nerve is derived from neural ectoderm, while the retina and retinal pigment epithelium are derived from neural ectoderm. Now for gene control, we'll start with PAX6. It's the master gene in eye development. It has a highly conserved function in amino acid sequence across multiple species. It plays a role in regulating melanogenesis, which is impaired in albinism. And mutations in this gene lead to the eyeless phenotype, first observed in Drosophila by Hoge in 1915. And on the right, we can see a Drosophila that has the eyeless gene ectopically induced on the wings and antennae. sonic hedgehog, SHH, responsible for the division of the eye field into two globes. Mutations are associated with cyclopia, one eye. It's important for developing the retina, fovea, and the positioning of our eyes. In wild type, the transcriptional factor Gli is upregulated as well as the receptor protein patched, while PAX6 is downregulated. PAX2, responsible for optic stalk formation and retinal fissure closure. Mutations lead to failure of optic fissure closure, which leads to no border between neural retina and RPE. Now for some paracrine signaling factors, we'll start with BMP4. It's secreted from the optic vesicle. SOX transcription factors are induced by this factor, and lens cells received BMP4. FGF8, it induces LMAF. LMAF is a transcription factor needed for the production of lens. And there's a diagram on the right kind of detailing that in Xenopus. Now for the disordered development, starting with the retina. Inner retinal migration is delayed and arrested early, leading to foveal hypoplasia, which can be seen in the diagram on the right. You can kind of see how the fovea has no pit and is flat. For foveal hypoplasia, no foveal pits, which is also caused by the retinal pigment epithelium not producing melanin albinos and having disordered structure. It's the single biggest cause of poor vision in albinos. Now for the optic nerve fibers, they overdecussate and cross up to 90% to the contralateral side, which leads to strabismus or cross-eyed. And as for the iris, there's little to no pigment due to the lack of melanin, which leads to transluminescence of the iris. And here's just a diagram of a pigmented and albino eye. You can see in the pigmented one, the pigmentation that's apparent, as well as if you look further down in the normal eye with normal production of melanin, there is not as much crossing between the retinal ganglion cell fibers and the chiasm. However, when you look at the albino, there's significantly more crossing. Now to speak about the retinal pigment epithelial cells. They are melanin-producing cells derived from neuroepithelium. They line the retina. Peak adherent is loosely distributed in albino RPE, which leads to the misshapen RPE cells. During invagination of the optic vesicle differentiates into two layers. The RPE and the neural retina, as we spoke about earlier, PAX2 is responsible for this. And when mutated, that differentiation does not occur, leading to no discernible boundary between RPE and neural retina. In mice, melanin synthesis in RPE starts at day 11 and a half in embryological development. Now, albino versus wild type RPE. And albino RPE possess defective shape. Fewer melanosomes are noted in albino RPE, therefore less melanin. OTX2 and MITF are transcription factors that are expressed similarly in albino and normal pigmented embryos. They're found in the nuclei of RP. OTX2 binds to bicoid-like motifs in promoter regions of the MITF target genes, such as tyrosinase, which is key to producing melanin. While these factors are present in albinos and wild type, the difference is that tyrosinase activity is diminished due to different genetic mutations, such as TYR or SLC45A2. I hope you were able to learn a little bit today about the development of the eye in albinos and in normal development. All citations are at the bottom of the slide in the notes section. Thank you. Hope you enjoyed.